So this is acrylamyl SE, the crosslinker, the anchoring reagent. And what this is a succedyl ester with acrylamide monomer attached to it. So this comes in a powder form, so you just have to resuspend it in DMSO or DMF, usually DMSO, at 10 mg per ml. Then aliquot it in 20 microliters aliquots, aerated with nitrogen gas, and then keep it at -80. Snap freeze it and keep it at -80. It's critical to actually snap freeze it and to deaerate it because the NHS ester hydrolyzes really fast. So it will expand the time this will last if you do it like this. Okay. So this is a 20 microliter aliquot, and what we're going to do is basically anchor our samples. So to do that, what I usually do is I have a very crude and simple standing box with a front plate of an STS page gel. I then get a bit of paraffin and a bit of water. So just put a bit of water here so that the paraffin sticks, and then you attach the paraffin, preferably without bubbles. And then you can do this in one of two ways. You either, so these 20 microliters will be resuspended in 2 mL of PBS. That's the recommend, but you can use more, also more concentrated than that. So because we only have, in this case, 6 cover slips, what I'll do is I'll resuspend this in 600 microliters, put 100 microliters in each place, and put the cover cell size down on top of it. You can also, if you prefer or you want to use more diluted or you don't like to put them cell side down, put a droplet on top. Okay? So either way works. Okay, so I'm going to pipette 600 microliters of PBS. You don't have to use fancy PBS like I'm using from Sigma, but any PBS will do, because here it's not very critical how pure it is or if it has dirt or stuff like that. Okay. So now I get my 200th, and the idea here is that these cells have been stained and permeabilized. So once I anchor them, you will attach acrylamide monomer to every primary amine available through the NHS ester. So it's critical that this step is efficient, because this is what will allow your sample to expand. If you don't have enough acrylamide monomers spread out throughout your sample, when you expand, the gel will not pull on your cells equally, so you will have artifacts. And you can do this, it's a classical NHS ester conjugation, so you can do this from as short as 1 hour to as long as overnight. Because you have, ideally, you want this to, most of the NHS ester to react, I usually leave it overnight at room temperature in a standing box, which is humid, basically. And that's it. That's the first step. Okay, so today what we're going to do is the gelation step. So after the anchoring, we wash the cover slips 2 times 15 minutes to get rid of any anchoring reagent that was in the sample, because basically what that will do, if it's not taken out, it will reduce the linkage between the sample and the gel. So I'm preparing the staining box. So basically I have, I think I showed this in the first movie actually. Pipette box, P1000, front plate of an STS plate gel, bit of water, parafilm, clean it, preferably with a bit of paper, so there's no liquid there. Okay, so this is ready. And now what it's going to do is we're going to prepare enough gelation solution for 8 cover slips. Because these cover slips are 25 ml cover slips, they're quite big. I prepared 200 microliters per cover slip. So, and I have all the reagents individually prepared, as in the protocol, and I always mix them fresh. Because the gelation is more efficient that way. If you're doing cells, you can keep them for about a week. If you're doing tissues, it's really essential that you prepare them fresh. Okay, so let's start. So I sent you sodium acrylates. The sodium acrylate, it's important that when you order your own, you immediately test if the solution is yellow. So as you can see, maybe, this is slightly yellow, ever so slightly, but if it's really, really yellow, that means that it's not good, and you have to order fresh from a new batch. Okay. So I'm going to carry on doing this. So I've also sent you I've also sent you acrylamide because it's very rare that nowadays labs have actually powder acrylamide due to its toxicity. So acrylamide. So this one was prepared recently, but I have one from 2016, for example, and it still works. So it's quite stable, has long, as it's prepared and it's stored individually. Okay. Same is true for the these acrylamide. I'm going to carry on pipetting everything. As you might be noticing, I'm not too worried about if the mixture is on ice now, but before when you start actually to prepare the final mix, it's important. Okay, so this is just a monomer solution that we're preparing now. Okay. Okay, so if you by any chance need to change the composition, because it might be useful, for example, to make it more concentrated, the mesh, you can increase the concentration of sodium chloride, for example, and you can also reduce the amount of water you add. And this might be useful for if you want to do more tightly packed structures. Okay, so we need PBS. Almost done. Sometimes just have to find it. There it is. So there's a lot of ways of doing this. This is just the simplest one. For example, one thing we have been trying to do now with a collaborator is to, rather than doing the digestion like we're going to do next, is to do the denaturing of the proteins, autoclaving the sample, and then staining afterwards. So this step is compatible with all of them. So this part is quite flexible. The only important thing is, after you anchor, if you do this, if you gelate it, you should progress with the protocol immediately. If you want to keep it in the anchoring step, because they're stable, and that's at that stage. Okay. Okay, so now this is the final components. Okay. So the next step is to divide these guys. Just steal the next part of the protocol, which is here. So this one has the details of the gelation solution, like it's in the protocol I sent you. The important thing is, for example, the APS should be fresh. I don't use it more than 5 or 6 times. I freeze it and thaw it every time. The TMAD is a 1 to 10 dilution, but that lasts basically forever. Okay? In the protocol, the way I wrote it is a bit more conservative, so it's a question of if what you're doing is more or less important. Okay. Okay. So I'm going to pipette the equivalent of 2 cover slips per tube, because you shouldn't do more than, well, you can, if you're really fast, you can do more than 2 at a time, but there's not really a big advantage of doing more than 2 at a time. Okay. And these guys, the individual ones, is what we'll keep on ice until they're ready to go. Okay, so now what I'll do, exemplified by this, I'll add the TMAD first, then I'll add the APS, and then I'll pipette the full 200 microliters on our staining box, and then I drop the cover slip on top, basically. Okay, so let me get a mock cover slip to show you. Okay. So if we imagine our cover slip, this cover slip was being washed, right? So what I would do is one of 2 things. I will grab it with some tweezers, for example. I would dry it very well and then put it on top. Or what you can do is also do a pre-wash with a monomer solution. So you prepare more and do a pre-wash so that it doesn't dilute the monomer solution there. And then you don't need to dry it, just drop it on top, okay? The problem with that is that if you wash it with monomer solution, you will have unpolymerized acrylamide, which is extremely toxic. So I avoid doing that. So when I dry it with a bit of tissue like this, the most I can, and then I just drop it right on top of the little droplets of acrylamide. You can try to make it more or less stable to cover the whole cover slip, but just the weight of the cover slip should be enough to make a perfectly homogeneous pad. So then we take this to 37 and we let it polymerize for 30 minutes. So we have our gelated cover slips. What I'm going to do, like I showed, cut this, cut this, just to separate them. Whoops. And then now they should come out easily. Yeah, if they don't, just cut it a bit more. Okay. So now I'll just get a second pair of tweezers. For example, these ones. And what we'll do is very carefully drop them in the place they're supposed to be. So What we want is for them, exactly, so what we want is for the cover slip to come off with the path, okay? Like this. Then we put them there, and then we basically cover it with digestion buffer. Again, the digestion buffer should be prepared fresh. Alternatively, you can do one of the other methods which do not involve proteinase K that are compatible with post-expansion staining. Those methods have the disadvantage of not preserving fluorescent proteins. This one, so the cover slip is breaking, so I'm going to gently take it with my hands, like this. Okay, so the advantage of this method is that it preserves fluorescent proteins, whereas the others don't. But on the other hand, if you want to stain very packed structures, you can stain them post-expansion, where the antibodies will have better accessibility. And basically that's it. After digestion, the pet should look something like this. This one has already been used, that's why it has the hole in the middle, but it's slightly enlarged and bigger than the 25mm cover slip. Okay, so to use it, you can actually grab the full pad and put it in water. And if you do that, it will expand more or less the size of this petri dish. Or you can also cut a small piece like I did here. As you can see, there's a small piece in the middle, and you can transfer that into water and expand only that. If that's the case, for example, you can also, so here you also see it very well, post-digestion, so you see the cover slip there and the pad much bigger and with a big hole in the middle where from the place where I actually cut a small piece to expand. So ideally you would do that, because if you can always, for example, keep this, whoops, you can always keep this in PBS, which is much stabler than keeping it in water, so you can reuse this pad several times. If you expand the whole thing, it will be less durable, because eventually the fluorophores will bleach in water. Okay, so you can always also shrink it back down by putting it back into PBS after it's expanded. But just be careful by cutting really round pieces so that it expands homogeneously and it comes back to the original size, which is homogeneous. Okay, so when it comes to mounting, what I do is usually this. Okay, so this is a dish which I have coated with PLL, and then I dry it very well. So either air dry it or actually dry it with a swab. And then I get a bit of the pad and put it right in the part where the PLL, being careful not to having any air bubbles. And then I put low melt agarose around it. Okay, so let me show you more or less how this would go. So we have our dish here. What you would do would be something like this. Let's see if I can put this like here. Yes. Whoops. Okay. Yes. So let's imagine I would cut a piece of the pad like this, probably more carefully than what I'm doing now, obviously, because I'm being a bit rough with it and they're quite fragile. Then you pick it up gently, okay, and then you get a piece of paper and you try to dry it the most you can without breaking it. Oh, and then you just carefully drop it
onto the PLL coated surface. I'll do that again because I think my hand was in front. So what you would do is this. So you just carefully drop it on top of the PLL coated surface. Okay. After this, you take all the air bubbles and you put low melt agarose around it. That's it. Then you put a bit of water on top and it's ready for imaging. Okay. So this time I didn't have a cameraman, so it's a bit more complicated, but I hope it was clear what I did. If you have any questions, do let me know.